Good morning. So finally we are on the day 5. You see, today we'll be talking in depth about that innate immunity, right? Some more concepts. But the second session, as I was not getting the time, so I thought that let's have the second session as much awaited session of that how to make your entire schedule for MBBS studies. So that's what we'll be doing in the second half, right? Okay. Today there is no exam, all right? Because what happens is that once we keep exam, so many students run away. All right, let's start with our today's session. Very good morning, and here we start. Say beginning is like this: immunity system, innate immunity response, and the adaptive immunity response. Today we'll be talking only about innate immunity response, that is which is non-specific. It means it is same for every pathogen, and at the same time, even if the pathogen is getting repeated, innate immunity has got no memory, right? No associated memory. So every time it will be behaving exactly the same way when it encounters even the same pathogen. But it is really fast. Within minutes to hours, it starts acting and it works quite efficiently. That's how we divided both the things into like this as a rapid action force, which instantly takes the decision, and while this adaptive immune system or the adaptive immune response was like our intelligence bureau, everything well planned. After the complete data is procured, then the planned decision is taken. Now, see, we'll talk about it in our second session when we'll be planning about, say, this MBBS studies. At that point, be ready for few questions. One of the question which I'll definitely be asking is, are you really planning to study and practice abroad? I mean, US. That means, in other words, simply asking, are you thinking of USMLE? Because see, if you are planning for that, then you have to really work hard and smart right from the beginning. It's not so that just after say 6 months, 8 months, 1 year, and suddenly you decide that let's appear for USMLE after a month. No, it has to be very well planned, and it's possible. It's not so that though it is considered as very tough exam on this planet, but doable, right? So more when we'll talk about it. Okay. Yeah, I was talking to someone about, say, it's not so that I'm just telling, but you guys have been exceptionally disciplined. Because I was say reviewing some of the videos I was watching, that hardly there was any comment which was off the track. So this is really very good. Okay, so here it is. We were talking about innate immune response, right? Non-specific, really fast, and no associated memory. So let's start with the action part. Some of the portion, it would be a sort of revision, but today what we'll be doing, we'll be going in depth about some portion. So it's like, say, if we have studied that this is the beginning, this is the end, right? And if the whole thing was divided into, say, 1, 2, 3, 4 stages, so that's what we covered yesterday. Today, we'll be going in depth about why this step 1 occurs. So that's the reason that immunity as such, if you look at it, they will be requiring minimum, I would say, 2 weeks. 2 weeks, right? Will be requiring minimum 2 weeks so that we can actually cover every aspect of it. That's why this time I'm thinking that after discussing with you in the second part about the MBBS schedule, right, what one should be doing, I have divided it in such a way that what one should be doing in the first 6 months, how one should be well prepared in the second half of the year, that is rest of the 6 months. Now, let's say, just for the example I'm taking, in fact, that is what is in my mind also, we go for respiratory system. But this time, when we'll be planning, and when we'll be starting our training, respiratory system once for all will finish it, right? We'll finish it. We'll not keep it like that, yes, you get the taste of immunity, you get the taste of, right, one structure after another, but it's like we'll start with respiratory and then we'll finish it. We'll take each and every aspect of it so that then you can say that, yes, respiratory system is complete. And then, as per the schedule, we can plan out that once your physiology part is complete, then we'll be going for the pathology of respiratory system. Because once you have understood physiology properly, then you are ready for the pathology. And by that time, some portion of the pharmacology will also be covered, so that you will also be involved into the treatment of that. Once this entire package is done, you'll be so comfortable, because that entire piece is what you have to send it for spaced repetition. That's what we'll be discussing, that what exactly is spaced repetition. And the discipline which you have shown in these last 5 days, right, same is what you have to follow strictly, because yes, no one will be there to take a follow-up, right? Though we can plan few exams. That's what I'll also be discussing in that it would be a sort of exam. Again, a very honest exam. But exams will be becoming slowly and gradually, they'll become tougher and tougher, right? Where you have to really think about it before answering it. So that's what is in mind, right? And today, you'll get one more flavor. Once the topic is completed, I'll be injecting few statements into that. Now, those statements, they might be from this topic, or they might be from some other topic but related to this topic. So what I'm trying to do really is that if this is one topic, so then my wish is to start creating that brain map, right? To start creating that brain map. As you'll become more and more habitual to it, any of the new subject, in that our notes would be only in the form of brain mapping. Only in the form of brain mapping. There'll be multiple brain maps. In fact, our entire training would shift to a very different, say, interface. There would be, everything will come in 2D and 3D, but only in the form of keywords. That will do when your brains are completely ready to digest that, right? Till that point, we are doing it like this, but slowly and gradually, you will be going for absolute brain mapping. The advantage is, at this point of time, you might think that, no, this method is okay. This is what really happens, because our brain is never interested in shifting. Brain feels very comfortable when brain is into the same scenario. That's the reason that many of the people, they say, no, we don't need promotion because we don't want transfer, right? They don't even take promotions, because in entire system, they say, where is the success? Say success is here. So then what is this? This is your comfort zone, right? This is your comfort zone. The day you are out of your comfort zone, you'll learn. This is what I really want. I want you to take out of your comfort zone. In every session, it should put some pressure on your mind that yes, there would be few questions. They will be tricky. I have to really think, I have to work. So that's why shifting from passive learning to active learning. This is a reason that I'll never be turning the chat off. Yes, you are free to write. But then when you write, you have to think twice that yes, whatever I'm putting over here, my words, they are carrying some importance, right? So slowly and gradually, in all the subsequent sessions, you'll start feeling that yes, even if everyone has answered, but my answer is this, right? Okay, so let's move back to our topic and more when we'll be discussing about. And yeah, tomorrow I think you must have noticed also there is a session and it will occur every Saturday at 9 p.m. Every Saturday. I have given the name for this week. The name is the Learn to Finish Champions Oath. Yeah, right. So in that, just its purpose is you discover yourself. Right, more will as we'll be talking about it. So here it is, the pathogen is there and pathogen attacks, right? He invades our immune system, steps in immediately. Macrophage, which are already into that tissue, they are the resident, they are our first line of defense, and they come for the rescue, right? They live in the tissue, and that's our local support. So within minute, that local police, it has arrived, and they start fighting. Okay, their purpose is they understand only one thing, that is hand-to-hand combat, right? Phagocytosis. They'll just go and they'll start eating up. They eat up pathogens and debris, right? So that this is the first line of defense. Our phagocytosis, it occurs because of macrophages, right? Macrophages, they are good, but the neutrophils, they are the experts. True, right? So they both work on the same principle, but only thing for neutrophils, we have to call them, right? So they come via blood supply. Now diving deep, pathogens have special molecules, and that's their signature. Here is a new word which is coming up, which we have not discussed till this point of time, that is PAMP, right? You'll be coming across quite a large number of abbreviated terms, right? This is PAMP. Now from this point onwards, it will always be called PAMP, pathogen associated molecular pattern. It means these molecular patterns, they are not present in humans. They are not present in humans. These are some special signatures, they are special patterns. So it becomes much easier that whenever this signature comes, actually the antivirus, right? The antivirus program in your computers, in your laptops, in your desktops, antivirus program, that is more or less on the same principle. The only difference is that that particular virus, which is in the form of a code, right? When we say, we call this thing as a virus, right? Why don't we call bacteria? Why don't we call parasite, right? It is called as virus only. I'm talking about computer virus because there is an interesting name associated with it, interesting full form associated with it. It is the vital information resources under seize. Seize means to capture, so it is VIRUS, right? Vital information resources under seize, right? That's okay. In that Three Idiots movie, the virus was something different. Okay. Yeah, I do watch movies and I do watch lots of movies, right? But once the work is complete. So this is the program which captures the vital information. Now, when these programs are written, right, there are specific codes. Those codes, they are nothing but like signature. So same way, exactly the same way over here also it is like that. These are the signatures, right? And what are they? What are they? Right? Pathogen associated molecular pattern. Which pattern are we talking about? Well, that pattern is when it comes to bacterial wall. So we have got peptidoglycan and LPS, lipopolysaccharide. These are bacterial wall. So we don't have in our body peptidoglycan or LPS. So as soon as these structures, these molecular pattern are detected, immediately it is known that he is invader. When it comes to fungal wall, there is Manan flagella proteins. And when it comes to virus will to go inside, so that's why it is the viral RNA or DNA. But in other words, PAMP is something, a pattern which is not welcomed into our body. If it is found, that means something is wrong, right? Same principle as that of computer virus. In fact, I many times wonder that in IT, most of the things, they are somewhere related to medical. Somewhere they are related. Even some names are also like that, right? Like say DNS, right? DNS is something which is in IT and it is in medical both. In IT, it is like domain name services, and in medical, it is like deviated nasal septum, right? Okay, all right. So now here it is our team alpha, right? Team alpha, neutrophils, eosinophils, basophils, and the mast cells, right? Neutrophils, they were like our fighters, those tiny fighters, right? Those who will be planning to sacrifice themselves. Eosinophil is our sumo, right? Big one, heavy. Basophils and mast cells, they are our snipers. Snipers means they are non-phagocytic. They will be operating from a distance, right? So they are non-phagocytic. Phagocytic. You got it, right? They are non-phagocytic. So they have this is our alpha team. So this alpha team should be able to identify that irregular or abnormal nuclear pattern, molecular pattern. So they have not nuclear, right? Molecular. So they have PRR, one more abbreviation. PRR is pattern recognition receptors. So now onwards, we'll start writing it as PR, and as we decided, we'll be writing receptor as this R. So this is PRR, that is pattern recognition receptor. Exactly, right? And this is for PAMP recognition, pathogen associated molecular pattern. Done? Okay. So we learned about this. Let's go into the types of it. The moment we'll understand this, it will make our entire understanding so easy that yesterday's topic will look like, yes, it is so logical and so simple. Because yesterday we understood one thing, that when these phagocytes come, they start attacking, right? How they attack, what they do that here is the pathogen, okay, gobble, 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 right? And then like that Pac-Man, right? They will gobble it up, and then phagolysosomes, they will destroy it, and done. So this was one thing. But there was no release of cytokines. What was cytokines? Cytokines was that like wireless message sending the information to the base that okay, we do have this type of attack, and then you put it into database. Now it is, if it is some minor attack, so then immunity will say, then why to trouble our headquarters, right? That's okay. Some pathogen came and we destroyed it. No need to tell anyone. In such cases, cytokines will not be released. However, if the attack is severe and in which he gobbled everything and then he said, okay, burst, boom, right? Sacrifice himself, because there would be a mass destruction of those pathogens. That is what is called as the oxidative burst, right? We talked about it. Oxidative burst. In such case, he said that because this attack was so severe, it is necessary to inform, and that's where the cytokines are released. Now, who controls all these things? How is it possible? Right? It is easier said than done that, okay, this is what really happens. Now we look at it. It is what is called as those PRR. Everything revolves around that PRR. Pattern recognition receptor. There are two types of receptor. So it means if this is happening, that means there is someone who is checking that is the attack severe or is the attack minor. If the attack is severe, so then let's signal, let's inform them. So here is the first type of pattern recognition receptor, which is called as the phagocytic. Phagocytic, simple. Pathogens, they are phagocytosed, right? Phagocytosed. So this is like pathogen and he is phagocytosed. But attack was minor. So no need to shout, right? No need to tell anyone. So simply cytokines, they are not released. In other words, it was, this entire process was minor. It started and it ended at that point only, nothing more. And yes, in the entire day, many of the processes, they do occur like this without even we noticing about it. But now, if the attack is major, right, there comes other type of PRR. Other type of PRR, which is like a signaling PRR. This signaling PRR will not be taking any part in any of the destruction or fight. Its name itself is signaling, right? Even in all those operations, there is one radio controller, right? His information, his job is to say supply the information to the headquarter and receive from there and send it to the troops. So here is, they help macroorganisms release cytokines. So small secreted proteins for communication, right? And that's what it is about, that it tells that this attack is major, send reinforcement, right? This attack is severe. So cytokines is where we know that attack has become severe and we require reinforcement. And this is the reinforcement which is demanded and it comes. Now, how come they operate like this? This is very interesting. The signaling PRRs, right? There are several groups, but the most important group, the group which is used always, it is called as the TLR, toll-like receptor. These are toll-like receptor, right? Toll-like. The name toll-like receptor, these receptor, they are like first single pass receptor. Now what's that single pass receptor? As we studied that whenever there is this phospholipid layer, right, cell membrane, there are few receptors which are like say 7-pass receptor. So here is the receiver and then 1, 2, 3, 4, 5, 6, 7, right? And here is the intracellular part and this is the extracellular part. These are single pass. So it's more or less like this, right? Single pass, single polypeptide chain. And this is found on leukocytes, epithelial cells, and endothelial cells. Now in TLR, remember TLR is what, right? Just keep getting, because now it will keep on getting divided and divided. So don't lose the track that what we are talking about. We are talking about, say, signaling, right? We are just talking about signaling PRR, right? Pattern recognition receptors. In pattern recognition receptor, one was phagocytic, right? Phagocytic, it makes logic, right? Just identify and start eating. But these are signaling PRR. In signaling PRR, this TLR toll-like receptor is one specific thing. Okay. Now these TLR, their function is to give signal. So if you are telling that we deploy one guard for the signals, right? His job is to send signal. It means he has to be present there, right? So over here, our purpose is to, that if this is the cell wall, right? Any information from the outside, so then there has to be a signal. Any information inside, so there has to be another person who will be handling this communication. So we have two groups. One group who is on the cell surface, that means outside. And there is another group who is inside the cell. Make sense, right? Now these numbers, total is 1 to 9. So 1, 2, then skip the 3 and 4, 5, 6. These are on the cell surface. Why come? They could have easily done it like say 1, 2, 3, 4, 5, right? That is on, say, on the surface, and then 6, 7, 8, 9 in the cell, right? They could have easily done that. Why to make things complex unnecessarily? Yeah, same thing. I also used to think about it, that I should have done like this if it was my chance, but it really happens like this. These numbers, they are not given in the order of their functionality. These numbers are given the order in which they were discovered, they were identified, right? So the way they were identified, they started giving the number. And we must thank them that they gave the number 1, 2, 3, 4, 5, 6, 7, 8, 9, right? They could have given some craziest number, craziest name, and then then our entire course would have been so difficult remembering the names only. Okay. So it was in the order which they were discovered. So 1, 2, 4, 5, 6. They bind to peptidoglycan. Remember peptidoglycan. Lipopolysaccharide? Yes. And flagella? Yes. And they were all what? All they were pathogen associated molecular pattern, PAMPs. है ना? Right? Peptidoglycan, lipopolysaccharide, flagella. And that virus part, we said that virus goes inside. That is virus RNA. So here, those TLRs who are inside, going inside 3, 7, 8, 9, they will bind to viral RNA. Completely logical. Right? So remember this part. On the cell surface and in the cell surface. Okay, moving further. 1 to 9, right? Whatever happens, any of the TLR, literally any of the TLR is activated, because normally they are into inactive state. They become active, right? They become active only in the presence of PAMP. True? Same way, right? If this is the girls' hostel, right? And if this is the road, you look at the boys, right? The way they drive the bike, right? So mare se, right? But the moment it comes over here, you look at the body posture, that entire thing changes. I'm not telling this, right? It was in one of the film where it was demonstrated that even penguins behave like that, right? So there was one very interesting shot. It was like one penguin, right? He was walking in such a mara style, and just after that small iceberg, he took a turn, and then there was a female penguin coming in. Immediately, his entire posture changed, right? Activated. So same thing, right? So in humans also it happens like this only, and so why should we blame, right? These bichare, these TLRs. TLRs are also like that. So they are activated by PAMP. Any of the TLR, right? When they are activated, so they activate. Now again, if there is any fight, right? Do remember this, because this time the opposition will faint. When you call him transcription factor, nuclear factor, kappa light chain enhancer of activated beta cells, right? He'll simply faint, and then you give a name NF kappa B, right? He'll keep on searching in all the dictionaries that what exactly that means, right? They will never get it. This is what they do. It would activate transcription factor, right? This transcription factor is a nuclear factor. It is defined in kappa light chain enhancer, and that is on activated B cells. Now it is making sense. B cells, they were not requiring that major histocompatibility factor. B cells are something which can instantly be activated. Immediately they'll be coming into action. So what is really happening to them? This is kappa light chain enhancer, right? So that's why our B cell is now called as the NF kappa B. This is what is getting formed. This would stimulate secretion of pro-inflammatory cytokines. Yes. So yesterday we were jumping straight away on this, right? We never talked about PAMP. We never talked about PRRs. We never talked about any of these transcription nuclear factor kappa light chain enhancer of activated B cells, right? We straight away said that when the attack is severe, they release cytokines. But actually the cytokine is released due to this entire mechanism. Now, what is the importance of this? Importance is that every point, if something goes wrong, there won't be any fight. And if there is no fight, that means that those pathogens, they'll keep on growing. And when they keep on growing, it would be converted from acute infection to chronic, right? And when it becomes chronic, that means their colonies, their population will keep on increasing, so much so that then the body's defense system will go wrong. When the body's defense system is no more capable, it will start invading even other areas. So that's the reason all these points, they are so important. Okay. So here is secretion of pro-inflammatory cytokines, and they are like TNF alpha, interleukin, right? Interleukin, IL-1, beta, IL-6. See, IL-6, nowadays, due to that COVID, many a times we are going for IL-6, right? Lab examination IL-6, and then when we find that it is raised, you know that why it is raised. We say that this IL-6 is high, that means that there is secretion of those pro-inflammatory cytokines. It means it is into the active phase. Now you know that if IL-6 is released, what really is happening in the background. So that's the meaning. Now once this is done, all those pro-inflammatory cytokines, right, they are released. They will lead to what? Vasodilation. Vasodilation, so then more leukocytes will come to the area, and you will find that patient is having fever. True, because now the battle is becoming more and more severe. And this makes complete sense. If the pathogen is intracellular, so cell would secrete interferon, right? So cell would secrete interferon alpha, interferon beta, because it prevents virus replication. See, in one page we learned so much. Now we know this thing, that when the pathogen is intracellular, that means in most of the cases it would be like a virus, right? When it is like a virus, so these interferon alpha beta, they will come into picture. But when there are, say, even on the surface also, if the pathogens, they are on the surface, so then there would be those extracellular PRRs will be coming into picture, right? 1, 2, 4, 5, 6. So that's how the whole process goes. Now let's think about life of that macrophage. What's really happening to the life of that macrophage? There is attack of bacteria. This part we know. And this macrophage has got PRR, pattern recognition receptor, right? So this macrophage would keep on searching and will say, okay, he has got PAMP. Now he is our intruder. So let's attack on it. First, he is arrested. He is arrested and he is put into a small jail, that is vesicle. This is what is called as the phagosome, right? That's what is called as the phagosome. That's it. So he is just put into vesicle, and this is phagosome. But then it is necessary to eliminate him, right? No case, no court, right? We just want to eliminate. Again, not my dialogue, that it was in some movie, right? And those lysosomes, they are called. And lysosomes, they are told that you open up over here and finish it. Now, when the lysosomes, they are into picture, it is called as the phagolysosome. Joint action, right? And in this phagolysosome, there are granules. Now see, once again, we are going into some details of it. Otherwise, yesterday we said that yes, these phagolysosomes, they'll open up, they'll release their secretion, which would straight away drop the pH. The entire thing is acidic and gone. That's it. We just discussed this much. Today, now we are going into one step, because we know that immediately associated with are some diseases. So that's what we'll be talking about it. So here it is, phagolysosome. It has got two types of granules, right? Because they are releasing granules into that cell. One is the specific granules. They are first to act. Another is in eosinophilic granules, but we'll talk about it. This is specific granules. These specific granules, say, all are AECs. They are enzymes. Whenever there are enzymes, so they will be active right? These are specific. One is the proteases and second is the hydrolases. Proteases and hydrolases, they are active at normal neutral pH, right, which is neither acidic nor basic. So they are first to act. All these enzymes, they require a specific pH to activate, specific pH to work on. So these are at normal. So bacteria is neutralized, almost killed, right? Not necessarily completely killed, but almost killed. There would be release of potassium and hydrogen. When this happens, into the phagolysosome, its pH value drops. When the pH value drops, there comes these azurophilic granules, right? These azurophilic granules. And they are severe, right? They are big boss. Cathepsin G, which is hydrolases, and this is myeloperoxidase, which is oxidative enzyme. They work nice in an acidic pH, right? Acidic pH. I think quite similar was happening when we were like kids, right? When we said that chal boss झगड़ा हुआ है तो क्या हाँ हाँ you go, you hit him, right? If anything happens, I am there, right? And then if they are beating us, so then we call kare come help, and then our those powerful friends, they used to come and save us, right? Same thing happens, right? Here is initially to we fought, but then we found that no, they are very powerful, right? So they are called cathepsin G and myeloperoxidase comes. This myeloperoxidase is the ultimate gunda, right? Oxidative enzyme, right? He is ultimate. When he comes, attack is neutralized, but this entire process is just 2%. Right? This is what really happens. But normal attack is not so light. This is okay for chota mota jagda. When the attack is not light, when it is heavy, so then it becomes a serious matter, right? In that serious matter, when the attack is heavy, so phagocytes, right, these neutrophils, they are the experts in that. They are experts. They are better even than the macrophages. Even macrophages, they are not that expert. Neutrophils, they are in real sense the experts. What they say, they gobble multiple pathogens and then it leads to oxidative burst. Yes, we talked about it, that it goes for oxidative burst, right? It is mass destruction but self-destruction too, and that's what really happens to those neutrophils. They sacrifice themselves. But in this oxidative burst, what exactly is this oxidative burst? There is production of nitrogen oxide plus reactive oxygen species. This name, reactive oxygen species, please remember this name. I'm putting a star. As in pathology, this name would come so many times. So many things will be associated with it, right? So you should not be lost in that where exactly are we. As this entire process is very vital, once we complete the topic, within 5 minutes, we'll be going for high-speed revision of this entire process. Now, in that process, I'm not telling you that you should be able to remember, you should be able to cram every word of it. Right? So this is like reactive oxygen species and the nitrogen oxide. They both are so important. Right? So during our revision, we'll be going very fast for all the steps so that that picture is clear. Then when you go back to the subject, right, you'll be able to go for those fillers. Fillers means if some piece of information is lost, again, in the next reading, that piece is filled up. That's what we'll be discussing into the second half. How should you plan it? Because we literally trust ourselves that yes, it is impossible to remember everything on the first stroke, right? Still, Our reading patterns are like that when we study, we revise it many times after such a long duration that only few contents are left out. Even in the second reading, it looks so new. So that's what we'll be planning into our say study pattern, that what should be done. Okay, so coming back to this, where were we? Oxidative burst, right? So in oxidative burst, this nitrogen oxide and reactive oxygen species, so there is production of it, right? And do remember this reactive oxygen species. Now this reactive oxygen species, this is chemically unstable molecule. Chemically unstable, right? This unstable molecule means he is always demanding. So what it will do, if it is having extra electron, so he'll give that electron to someone who is stable and will make him unstable. Or if he is deficient, he'll take that electron. In either case, he will make the opponent unstable. When the opponent is unstable, it is liable to break. That is what is called as tissue damage. So over here, this superoxide anion or hydrogen peroxide is very powerful, very powerful. They will bind to molecules within the cells and they will lead to tissue damage, right? But again, both are destroyed. So pathogens and neutrophils, or pathogens and macrophages, whosoever is acting, but that's okay. We said that neutrophils, they are experts. Yes, they are experts. But even macrophage, they also work on this part. But very important point in this case is this. Sorry. I'm putting 2 stars. NADPH oxidase is used in the process of oxidative burst. Now there is a whole mechanism for NADPH oxidase, whole mechanism. Right now we are not touching it because it's not so that today is the last lecture of immunity. In fact, now when we'll be planning the series of immunity, for 1 week it would be only immunity, but then these sessions, they will be considered as base and straight away we'll be mounting on it, right? So by the end of 1 week, this entire part would be fully completed. So that's about future, right? We'll be planning it. But over here, we are just adding, we are injecting a new name into this. This is NADPH oxidase, right? NADPH oxidase. This is playing one important role into oxidative burst. It means in case if NADPH oxidase, right, it is not present, something goes wrong. So now this bacteria would escape from this killer cycle, right? He'll be saved. Once it is saved, so he is not killed and he'll keep on developing, making bigger and bigger colonies. So that acute pathology, it goes into the chronic pathology. Chronic pathology. So this is what is called as the chronic granulomatous diseases, CGD. So that's the reason. It means one single thing, any DPH oxidase is not present and it leads to this, right? Why it should happen that any DPH oxidase is not present? Because see, any DPH oxidase, it will be requiring certain environment. As we discussed, that some say enzymes, they are working at neutral pH, some enzymes, they are working at the acidic pH. So if this environment is disturbed, they'll stop working. Then there could be some drugs, right, whose side effect could be like this, that they are inhibiting NADPH oxidase, and that leads to formation of so many things, right? So they are called as the side effects. Side effects. It means here is drug A, and drug A could be good for, let's say, cosmetics, right? It is some good drug for cosmetics, for the beautification. But who knows that at the same time it is NADPH oxidase inhibitor. So it means nothing would happen till there is no infection. But the day there is some infection, anything goes wrong, this is the trouble. This is what really happens, right? Yeah. Now these are some very vital concepts. Some vital concepts. The TLR part, toll-like receptors. Now I am putting few words. When talking about receptors, we said that these are called as extracellular ligands. Ligands means nothing but simply some chemicals, right? Some drugs, some toxins, some hormones, some enzymes, right? All those things. When they come in contact with them, there is some change, right? There is some biological response. So these toll-like receptors, they interact with extracellular ligands on pathogens plasma membrane. Now see these pathogens, they are super smart. They are also knowing. Now it is like, because we are on our side, it's like if our team is playing, right? So we always feel that yes, India should score 1,000. That's what I used to think, right? I was very optimistic. So when I used to watch those cricket matches, and I used to keep calculator that okay, India is requiring 120 runs to win, right? And we do have just one wicket left out, and we do have 4 overs to go. So then I used to calculate like okay, 36 runs per over. So that means we do have chance of 144 runs in next 4 overs. Okay, no, still there is hope, right? Then as one by one the ball used to decrease, so my tension used to started in now just in this ball there should be 100% a six, right? And by chance if it happens, so then I used to say okay, fine, so this much work is done. Now we have to go for only 114 runs in 3 overs and these many balls, right? Well, mathematically possible, not practically, because we always think from our side. For time being, think from the pathogen side. Let's say we are pathogens. So then, will we be going with such ease that okay, theek hai, let us be sacrificed by those neutrophils, and in case if we get the chance, will infect the body, otherwise will be sacrificed? No, they will not do like that. Pathogens, they are also super smart. They know that this is going to happen, so they are also well prepared. So now for next few points we have to think from the pathogens aspect. That's why I see in one of the very nice the detective serial, they say that if you really want to be a good cop, think from the perspective of the criminal. You should be having the criminal psychology if you really want to become a good cop. And that is when we'll study forensic medicine. In forensic medicine, which is my favorite, because then everything is associated with one case, right? Everything. And actually when learning forensic medicine, right, don't tell anyone, well, I should say it or not, but well, we can plan very smart crimes, right? Doctors, they can plan very smart crimes, because if your knowledge is good, so then you know that what would really be happening into that autopsy, what would really be happening, which findings will they be going for. So if you are read in the habit of reading Robin Cook, he is an ENT surgeon. All his books, those books, that description itself is so fascinating, right? For first 2-3 pages, right, it will be that description will go like that 33.7 grams of cylindrical metal, as it pierces the first layer, and then smashing it into the small molecules, and as it touches, and then the entire description will go on, and when it hits the wall of that tough structure and then the cortex breaks, it means he's talking about a bullet which has been fired, and then it passed through all the soft tissues, it hit the bone, and then the bone is shattered. But that description would go on for say 3-4 pages. But if you start reading it very scientifically, it is so interesting. Every means entire thing, any of the book you pick up, right? Whether you take mutation, whether you take virus, any of those, and so all those books, right? They are having this amazing description. So in forensic medicine, though, we'll be learning forensics like that only, just in the form of case, so that we don't have to remember. Okay, coming back to where were we? Okay, so we were over here, we're talking on the about the ligands, right? So these are receptors. Receptors are obviously activated by the ligands, extracellular ligands, and these are single pass, right? They are single pass. So anything happens over here, it will be having effect inside on pathogens, plasma membranes. True. The second point is, yes, we remember, right? I suddenly got remembered that it was like this, right? That pass my message inside. Yes. The mucus secreted by mucosal epithelium, right? It traps pathogens. So this mucus secreted by mucosal epithelium, it creates an environment, it traps pathogens, and this is part of innate immunity. That's something which is already present. Now comes another interesting portion, very interesting portion. We talked about pathogen associated molecular pattern, right? Imagine if you are an assassinator, you are given a task that, okay, knock off this person, and you are on the hunt, right? Whether you are a sniper or you are expert in hand-to-hand combat, you finish the job. Once you finish the job, how will you report back? What will you do is you will say, okay, work done. But then, because you will be given the money for it, how to trust? So you have to send a proof that, okay, this is done. Same way over here, there has to be a damage associated molecular pattern. When our attackers, when our soldiers, they know that, okay, yes, the pathogen has been neutralized, it means we declare that enemy is dead when we get DAMP, damage associated molecular pattern. So when we have damaged the enemy, then it is at that point this DAMP is sensed. Who will be sensing it? Same, our PRRs only, right? Our pattern recognition receptors, they will sense both the things. PAMP, they'll say that yes, enemy is alive, attack on them. And then when they are damaged, that is damage associated molecular pattern. So now we know that it has been done. So that's how it will work accordingly. Okay, now stop. Otherwise, they'll keep on fighting. They'll keep on working. So this is what is also called as the negative feedback mechanism. There are two types of mechanism which will always be going into our body, because if we start anything, it has to be finished also. And we have to stop at certain point, right? Otherwise, if the process will keep on going on and on and on, it leads to what's called as the burnout. It is what is called as the burnout. This is one of the topic which we'll be discussing when we'll be planning your MBBS studies, right? Just after an hour or so. Because I don't want that you should be burnt out. Yes, initial enthusiasm level is so high that yes, you will keep on pumping 5 hours, 6 hours, 10 hours. Yes, it would happen. But then in a long run, is it possible? Well, there are few extremely brave people, right? They consistently read with that much dedication throughout the year. right? But honestly, not me, not me. And I know most of you are like me only, right? That there are so many good things in life, right? Is it only this study? And what you said is that yes, you were missing those snipers, right? You were into those vibes of PUBG when you were such a good sniper, but man, now you are a much better sniper because now your level has increased. You are not talking about those basic snipers. Now you are talking about those finest of the snipers which are into our body. You are talking about basophils, you are talking about mast cells, right? Much smarter snipers. True? So that's the thing. Okay. The fourth point is these PRRs, they identify PAMP, right? This is just to remind you. PLR, toll-like receptors, they are family of PRRs, and they have how can they really identify? Again, we are putting something extra, right? This reminds me of that Paresh Rawal when in that movie he said, अच्छा तुमने फिर से problem बदल दी. Right? So dimers of chains with extracellular leucine-rich domains. Right? See such names, right? So it always fascinates that if someone fights with a doctor, right, how nicely we can tackle that non-medicos, right? By using such words, they'll be knocked off. What TLR? They have dimers of chains. Right? Let's make it simple. There is extracellular leucine rich domains. So one thing is that's the keyword leucine. So there are leucine rich domains. Domains means these are the areas. These are the areas which are having leucine. So this means it is this leucine which will be going and binding to those microbes, right? So this is the leucine which is important, leucine rich, and they are extracellular. And there are dimers of chains. So it's like a complete chain in which, say, these leucines, they are hanging, right? So, and these leucines, they will go and they will stick to those microbes. Now, that's what our TLRs will be doing. But our pathogens, they are not that simple. Yeah. Oh, it's buffering? Really? Can you hear me? Is guys, just type something. Just give me the message. Is everything okay? Can you hear me and see me properly? Or it's buffering? Is it okay? Uh, hello, hello, hello. Right? Is it okay? Hello. Everything is all right. Oh, okay. Good, good, good. Nice, nice, nice. Okay, okay, okay. So we were talking about that those leucine rich. Okay, okay, stop. So those leucine rich domains, they'll be coming and they'll be giving a handshake to these microbes. They'll capture them. But then our pathogens, they said, we don't want to talk to you. Right? Why should we? So they are extremely slippery, extremely slippery. Now, such slippery surface, right? Say if it's like, say, bowl of ice cream, right? And you try to eat that ice cream with the hand, right? How slippery it is. Chances are so much that that ice cream, that delicious ice cream would fall, right? But then we are smart. We come up with a spoon, right? Or if it is rasgulla or anything, right? I know, right? I'm speaking because it is festival time. So all those things, they are quite slippery. So if we really want to eat it, we have to either use a fork, we have to use a spoon, right? We just cut it and then we eat it. These pathogens, they have extremely, right? They have got extremely slippery surface. So it is not very easy for even our all those TLRs to act on it so that they can slip off during an attack. That's what their intention is. Because if the surface is slippery, before they can act right, they will slip off. That's how some of the pathogens, they go, actually, they reach all the way up to the lymph nodes, right? So there has to be a mechanism. Here is, we are coming up with a new word, and this word is very, very important. In fact, in future, we'll be talking about this for almost an entire session. It's what is called as the opsonization. Yeah, remember the word. It's called as the opsonization. What opsonization is? So these are the soluble proteins attached to pathogen surfaces. Right? Soluble proteins. So say for example, let's say this is a very powerful tank, right? And when we really want to attack on it, so then, some of the bombs, they just stick on it and then they explode and they create an impact inside and the entire system is affected. Same way over here, we know that this entire surface is so slippery that there is no point that the whole thing can be engulfed or it can be destroyed. They create some areas, and those areas are like those soluble proteins, they are attached to pathogen surface. So these are our areas, and it is easy for phagocytes to identify that yes, this is where we have planted our soluble proteins. Go and stick to that. So now it is possible to catch hold of this pathogen, and engulf it, right? This process, which we have described in just few steps, but it is what is called as the opsonization. Keep the word in mind. Soluble proteins, they are attached. Soluble proteins which are attached to pathogen surface. So that these soluble proteins, they are identified, and that's how they go. The next one, we talked about CD, right? Cluster differentiation, right? Those cluster differentiation. There are several numbers. Sometimes if we said that there was CD4 plus, then CD8 plus, right? So CD4 plus, he was just giving orders, right? He was sending that, okay, B cell, you are promoted to plasma cell, and now you have got a heavy machine gun, and then start firing. CD8 was telling, no, no, no. Now I have also got the Josh, right? I'll also start firing, cytotoxic, etc. All are having numbers. This is CD27. This CD27 is like a white flag. He'll say, when this cell type comes, he'll say, okay, no phagocytosis, please, right? This is like ceasefire, right? No phagocytosis. When he comes up, he's like a white flag. Because our neutrophils, they are not understanding that sometimes out of over enthusiasm, they'll start killing their own cells. And that's where many of the pathologies, they occur like that. Because if there is some mutation, and then this particular area, right, if it mimics like pathogen associated molecular pattern, so our immune system is completely fine. They'll say, Kacha, these are pathogens. They'll say, no, we are not pathogens. We are the normal human tissue. We are your own side. But they say, no, you are wearing that dress. Okay, attack. That's where this thing will come into picture. He'll say, okay, CD27, see, here is the order. We were given the special instructions to work like this. So then they'll say, okay, now we'll not attack on you. Right? These are the cell types. CD27, remember, very important, right? When we'll be discussing about autoimmunity, etc. The next important point is that most of the cell types of innate immune systems, they are phagocytic. Most of them, they are phagocytic, except those two snipers. Otherwise, most of them, they are phagocytic. And when we say, we use the word snipers so frequently, right? When we say it's snipers, Why we say snipers? What will they be doing? They release what? What do our snipers, that means our basophils and mast cells, what exactly they release? Don't worry about what everyone is typing, just you type your answer. Don't look at others' answer. And this is like a question. What do basophils and mast cells secrete? Just type your answer. Don't worry about what everyone is typing, right? Whatever you think, write it down. It's like in a wave, answers should appear, right? Don't worry, don't wait for others. Okay, targeted receptors. What do they secrete? See, basophil and mast cells, they secrete histamine and pro. That's okay. They do their actions from a far distance without hand-to-hand combat. That's okay. That's what sniper is about. Yes, this is the thing. Histamine and pro-inflammatory mediators, right? Pro-inflammatory mediators. That's actually the answer, right? We remembered that sniper is like, just for the understanding. Actually, I also did that thing once, that when in one of the viva, when it was asked that which are the 8 bones over here, and my answer was, sir, she looks very pretty, try to catch her. Sir said that it's okay, this is how you remember it, but what are the real names, right? So in exam, don't use the word snipers, because that's, yeah, the answer is histamine and pro-inflammatory mediators. Okay, all right. Moving on to the next point. This is type 1 interferons. Type 1 interferons, they are produced by virus infected cells. Just remember this. Type 1 interferons, they are by virus infected cells. This is easy. Response time of, say, innate immune system is in hours. Right? It is pretty fast. Yeah. Now this is chemokine, one more new word which we are now talking about. One more new word. Chemokine. This is a type of cytokine. So what was cytokine? You remember, cytokine was like a message which was sent to other cells that send reinforcement. In that reinforcement, we do have several type of messages, because if you are handling the communication control, so it's not only one type of message, there will be several types of message. Same way, you have got cytokine, but this chemokine is a type of cytokine. That's also called as the interleukin 8. So what this interleukin 8 is, this is chemoattractant for neutrophils. So in early stages of infection, if that query comes in your mind that it's okay, macrophages, they were acting locally, है ना? Macrophages, so they live into that tissue. So as soon as there was attack, immediately macrophages, they come into picture and they started working. But when that attack was becoming severe, so at that point, that reinforcement was needed. But in the process, by the time others come, right, those sumo and everything, that is our eosinophil and everyone comes, right, our snipers, they arrive at the scene, right, even other cells when they jump into the scenario for that battle. Before that, let's call in the early stage those neutrophils, because those neutrophils, they are actually the experts. So let's call the experts fast. It is at that point when this chemokine is released. This chemokine, sophisticatedly called as interleukin 8. This is nothing but a type of cytokine, but it tells only neutrophils that you come fast. And they'll say, where should we come? They'll say that wherever you see that yellow smoke, right, come. So this is like, again, that smoke bomb, which will telling it as a spotter. So this chemoattractant or the spotter, right, when that is released, immediately those neutrophils, they will come into picture. So that's the whole idea, that it is the chemokine which is chemoattractant for neutrophils, but in early stage of infection. Because in late stage of infection, so there is entire troop which is coming, right? Everyone is there, right? Or everyone has started, let's say, joining the battle. The next point is respiratory epithelium. In respiratory epithelium, yeah, there is one very nice concept, surfactant. When we'll be discussing, say, respiratory system, that is physiology, again, surfactant would be our guest of honor on the day when we'll be talking about, say, the normal physiology. Very vital concept of this surfactant, because that surfactant is something which is keeping all the lungs properly inflated, and that's how systems they work, right? Respiratory epithelium, where there are surfactant, it blocks bacterial surface to prevent infection. Yes, it is like an innate immunity, right? It is preventing bacterial surface. It blocks bacterial surface, and that's how the infection is prevented. So it means those, from the pathogen's point of view, if you have to define, right, if you have to decide or you have to define any of the attack, you will also be taking care that yes, it is the surfactant where there will be the trouble. So we need to break that surfactant also. And that's where all this smoking, right? Why smoking is so bad, right? It is because it is actually destroying such amazing systems which God has already given to us. These are the natural, very fine defense systems, and by using all these bad things, we are destroying ourselves, right? So that was for today. Now what we'll do, we'll go for a super high speed revision of this entire topic, right? We'll be going pretty fast, right? So here is back to good morning, and here we start. This time, what you should really do is you also start speaking, right? You also speak along with me. I'll be marking those important keywords, so you also speak. When you speak at such high pace, you'll be surprised that you'll start remembering so much. That revision itself will become very easy. What you have done is you have deployed more number of senses, right? And when actually you must have seen that when I revise, I draw like this, I sometimes draw like this, I make star, right? Because when you are involving any of those symbols, right, it is enhancing your learning ability. Okay, here is, we start with immunity system. We talked about innate immune response, right? That's what we shall be discussing today. Second one was adaptive immune response. This innate was non-specific, same for everyone, really fast within few minutes to hours, but there is no associated memory. So it will work every time in the same way, even with the same pathogen. Let's go deep. Here is the pathogen attacks, right? He attacks, immune system immediately steps in, and because it is the macrophage who is resident, he is living into the tissue, and it's like a local support. So within minutes, immediately he'll come for the attack, right? Here is macrophage or neutrophils. They are our phagocytosis. So they eat up pathogens and debris, and that's why they are our first line of defense. Going deep, pathogens, they have got spatial membranes or spatial molecules, right? These are the spatial molecules. They are their signature. These signature, they are not present in the humans. So that's why these special signatures, a name is given which is associated with only those pathogens. So that's why they are called as pathogen associated molecular pattern, right? And these pathogens, they are of several types, whether it is a bacterial wall or it is in fungal wall or it is inside the virus. On the bacterial wall, we have got peptidoglycan NLPS, that is lipopolysaccharide, right? In case of fungal, it is mannan and flagella protein. Viral, viral RNA, DNA. Here is our team alpha. Team alpha, they are quick to react, and they were like neutrophils. They were our small fighters, right? Sacrificing themselves. Eosinophil, which is for those gawky parasites, big parasites, huge parasites. That's why their size is big, because they can eat big. Basophils and mast cells, they are non-phagocytic, right? So that's why they'll be like snipers. They'll be releasing only histamine and pro-inflammatory mediators from far, no hand-to-hand combat. Now all of them, they have got the PRR, right? Pattern recognition receptors. That is for identifying PAMP, that is pathogen associated molecular pattern recognition, right? That's what they try to recognize. These PRR, they are of two types. One is phagocytic, pathogens phagocytosed, but attack was minor, no need to shout, cytokines not released. So these are the PRR, they identify, they reach, they kill, khatam baat, right? Others, they are like signaling. If the attack is major, so they need to tell, they need to call for help, and that's where the cytokines, they are released. Cytokines, they are small secreted proteins, and their function is only the communication, right? And they say send reinforcement, right? Now these signaling PRRs, if we go still in deep, what are they? They are like, there are several groups, but one of the very important, most important group is TLR, toll-like receptors. These toll-like receptors, they are not 7-pass, they are single pass, right? So it is like this, single pass, right? Which are those 7-pass? They are like GPCR, G protein coupled receptors, right? They all are 7-pass. That is GQ, GI, GS. You remember? We talked about it. Okay, so single pass receptors, this is single polypeptide chain that is found on leukocyte, epithelial cells and endothelial cells. And in this TLR, once again, we super divide them into whether they are on the cell or inside the cell. On the cell, so it is 1 to 6 except 3. So it is 1, 2, no 3, 4, 5, 6, right? So that's how we remember it. And they bind to obviously all PMPs. So peptidoglycan and lipopolysaccharide, that is in case of bacteria, and flagella in case of that fungus. Within the virus, it has to go deep. So that's why our one of the agent is inside the environment, and that is having the code name 3789, right? He'll bind to viral RNA. Whatever all of our agents 1 to 9, we get any message from them, they will all be talking on the same language, and that is when they are activated by PAMP. That is when they are able to identify PAMP, they will activate and they lead to transcription factor, nuclear factor, kappa, light chain enhancer of activated B cell, NF kappa B. What it really does, simply, it is secretion of pro-inflammatory cytokines. This is what is done, right? TNF alpha, interleukin 1 beta and IL-6 leads to vasodilation. Vasodilation, so more leukocytes, they come, leads to fever, right? And if we are dealing with pathogens which are intracellular, right, intracellular, then cells secrete interferon alpha and beta, and that prevents replication of virus, right? Though there are so many other methods also, but at least the function of this interferon alpha and beta is to prevent virus replication. Moving on to the life of macrophage. This macrophage, which was associated at a local level, he was live locally, right? Attack of bacteria, and he will sense from its PR receptor, pattern recognition receptor, and he'll say, okay, you are there, all right, arrested. And he is put it into a small cubicle. We'll name it as vesicle, and that's name is Phagosome, because he is put into some jail, right? So phagosome. And then comes those lysosomes, right? They are the other gundas, small chota gundas. They are sent and they say that finish him, right? They will go there and they will start releasing the toxic gases, right? That's how the entire conspiracy is done. Phagolysosome, that is the name which is given, and they finish this thing. But then These lysosomes, they say that we do have two types. Granules, two types of granules. One is the specific granules. First granules, they are to act, and they have got proteases and hydrolases. So when everything is fine, neutral pH, they simply go, they finish the work. Bacteria is neutralized, killed, and it will release potassium, hydrogen, right? And then into that phagolysosome, now the pH is dropping. When the pH drops, here are the bigger fighters. They'll say, is there anyone left out, right? Here is azurophilic granules. They have got cathepsin G and myeloperoxidase. These are very powerful hydrolysis and oxidative enzyme, but they work nice only on acidic pH. And now the pH is acidic, so clean them, right? And everything is done, neutralized fully. But this type of attack is only in 2% of the cases. Still, the bigger attacks, they are left out. Normal attack is not so light, it is heavy attack. So phagocytes, right, these neutrophils which are expert in that, macrophages are also there. They gobble multiple pathogens, and then oxidative burst, boom, right, gone. And it is a big damage, mass destruction, but in the process also it gets destroyed, right, himself also. What it really is doing, there is production of nitrogen oxide and the reactive oxygen species. That's reactive oxygen species. These reactive oxygen species, they are dangerous due to the simple fact that they are chemically unstable. If they are unstable, they'll make others unstable. And these are superoxide anion and hydrogen peroxide. Hydrogen peroxide being extremely powerful, right? What they say, they bind to molecules within the cell. When they bind to those molecules, they are unstable, they'll make that cell membrane also unstable, and that is what leads to tissue damage, right? But both are destroyed in this process. This entire process, still if we go into more depth, we'll come across one substance, that is NADPH oxidase. This is one of the very vital substance, right? And that is used in the process of oxidative burst. So by any means, if this NADPH oxidase is affected anyway, right, whether it is because of some toxicity or it is because of some mutation, anything, so then the bacteria really escapes. And when the bacteria escapes, that acute condition, it goes into the chronic condition, and that is what is called as the CGD, that is chronic granulomatous disease. Okay. Then those important vital concepts, they were like, that is in TLR, right, it is the toll-like receptor interact with extracellular ligands because they are receptor, right, on pathogen plasma membrane. Mucus, very simple, right? Mucus, it is like first line, right, basic line, it traps pathogens, and that's the part of innate immunity. The way we have got PAMP, pathogen associated molecular pattern, when they are alive, when they die, so here is the proof that we have finished our work, and we sense DAMP, damage associated molecular pattern. So all good, right? We have finished the work. And then we have got the PRR, they identify PAMP, that's right, we know this. And in this TLR, it is the family of PRRs, and they have, that's a word, leucine. Those leucine, they are like those hanging areas which will be going and will be binding to microbes, and all of them, they are connected with dimers of chain. So the dimers of chain, and then that extracellular leucine rich domains are there, that the area, and which will be going for the binding of microbes. Then pathogens, here is the smart surface. It is a slippery, it is extremely slippery surface. So we must know a process, that's what is called as the opsonization. So it is the opsonization, that's the solution for taking care of that slippery surface, because otherwise bacteria will slip off during the attack. In order to do this, we'll be utilizing some of the soluble proteins. So let these proteins be soluble on the surface, and when that is there, so they'll be acting as our helipads, where our helicopters will go, will land, and will place the entire bombs, right? So this is easy for our phagocytes, our team, to go over there and act, right? And because they are, those areas, they are planted by us, so it is easy for us to identify. Then we don't want to destroy our own areas, so that is where will be utilizing CD27. So remember, this is a code. This is a code. CD27, that means don't eat us, right? We are amongst you only. Most of the cell types of immune systems, they are phagocytic, except those snipers, that is the basophil and mast cells. Type 1 interferon, that is released by the virus infected cell. Response time, most probably in cases of innate immune system, that is very fast. It is within few hours. This is chemokine. This is for attraction, right? It is for attraction, chemo attraction, chemical attraction for neutrophils, right? Those who are the experts into this early stage of attack, right? So they are the chemokine. Chemokine are nothing but they are type of cytokine. Cytokine is something which is used for the communication between those cells, right? That's a communication link. Respiratory epithelium, it has got the surfactant, which is important concept for the respiration, right? And the lung condition. So it blocks bacterial surface. It is not permitting the bacteria, right? And that's how it prevents the infection. And that's it for today, right? So just after about, say, half an hour, right, we meet again and we shall be talking about your entire MBBS planning, right? We'll cover up many of the important aspects. Yeah, Telegram, I'll definitely be we are doing it by this weekend, right? Saturday, Sunday. So I'll surely do it. Right. All right. Thank you so much. And we meet very soon. Bye bye. Yeah. Yeah, Insta ID also. Sure. Thank you.